The Krebs cycle is the second of the two steps in cellular respiration. In the first step, glycolysis, glucose was broken down into two 3-carbon molecules called pyruvic acid in the cytoplasm of the cell, indicated here by a big yellow square. And in breaking it down, cells are able to retrieve a little bit of energy in the form of ATP and NADH. And they can do this in the absence of oxygen. But if oxygen is present, cells can get a whole lot more energy by breaking down those pyruvic acids to retrieve the energy from them. This can only happen when oxygen is present, and it happens in the mitochondrion. A cartoon of the mitochondrion is drawn here, and I'm just going to give you some anatomical names so we can talk about this mitochondrion with the same terms. The outer membrane wraps the mitochondrion, and inside that mitochondrion is a second membrane, and that second membrane is actually larger than the outer membrane, and because it's larger and it has to fit inside the smaller membrane, it's kind of crumpled up the way a plastic bag might be crumpled up in a smaller bag. And this is, of course, the inner membrane. There's also a space on the interior of the inner membrane that is interesting. Now, in photosynthesis, that space is called the stroma, but in the mitochondrion, that space is called the matrix. And then finally, there's a space we're going to talk about that is functionally similar to the space inside the chloroplast's thylakoid, and that is the space between the inner and outer membrane that we are going to call the intermembrane space. Okay, so these are the 4 areas that are of interest in the mitochondrion for the purpose of talking about cellular respiration. All right, so let's focus on the Krebs cycle. The Krebs cycle is the first of 2 steps that happens in the mitochondrion. And in the Krebs cycle, the pyruvic acids that were produced by glycolysis get imported one by one into the matrix of the mitochondrion. And there, each carbon from the pyruvic acids is popped off one by one and converted ultimately to carbon dioxide. Since there are 6 carbons in those 2 pyruvic acids, eventually we produce 6 carbon dioxides, which get released into the environment. So for you, what that means is that you exhale them. So when you eat glucose, you exhale that carbon waste. Okay, so let's talk about the Krebs cycle in a little more detail. What we're going to do, instead of following both of these pyruvic acids at once, we're following just one of them. And then when we're done, we're going to multiply everything that we do by 2, since there are 2 pyruvic acids. So let me take you to a drawing of the Krebs cycle. And it looks like I've gotten all sorts of mess all over here, so I'm going to erase all of that and do it again. Okay. Here's our little pyruvic acid molecule, and I'm going to label it. So we just have one of them here. And remember, when we are done, we're going to multiply everything we do by 2. Okay, so the first thing that happens is that this pyruvic acid gets imported through both the outer and inner membranes until it's in the matrix. So here it is now in the matrix. And there, the cell is able to remove a carbon dioxide. So one of the carbons that was on the pyruvic acid leaves as carbon dioxide. And as I said before, whenever the cell removes carbon dioxide, it's able to harvest energy. And it harvests energy in the form of 2 electrons stored in NADH. That leaves us now with a 2-carbon molecule that still has pretty high energy in it. Pretty high energy. In fact, it's so much energy that the cell can't break it in two. Because if it were to break it in two, it would release so much energy at once that most of that energy would be lost to the environment in the form of heat, and the cell would not be able to capture that energy in molecules like NADH or ATP. So, what the cell has to do to control how much energy it removes and do it a little more slowly is attach this 2-carbon molecule to a larger molecule. So that's what's going to happen in this second step. Our 2-carbon molecule is going to get attached to a 4-carbon molecule that's always there in the matrix. So I'm going to call it a 4-carbon resident. And this 4-carbon molecule has very low energy. So whatever energy it has, is the energy it's going to pick up from that carbon-rich 2-carbon molecule. So we're going to attach our pretty high energy 2-carbon molecule to our low energy 4-carbon resident molecule. All right. Turns out that's hard for the cell to do. So in order to make that reaction happen a little more easily, an interesting thing happens. It attaches to the 2-carbon molecule an additional little molecule called coenzyme A. Abbreviated as CoA. And coenzyme A acts like a glue stick. Helping attach that 2-carbon molecule to the 4-carbon molecule. So now, with the coenzyme A attached, the 2-carbon molecule gets attached to the 4-carbon molecule. And here you can see that those two molecules are now stuck together. Once coenzyme A has done its job, it lets go, and it can be recycled to do this job again and again and again. Now we have a 6-carbon molecule that has all the energy from that 2-carbon pretty high energy molecule. And we're going to spend the rest of the Krebs cycle removing that energy to restore that 4-carbon low energy molecule. Okay. The reason it's different than our 2-carbon molecule is that instead of all the energy being locked in the very few bonds that are holding the 2-carbon molecule together, that energy is spread out across all the bonds in this much larger molecule. And what that means is that when we remove 1 carbon, we're only removing a smaller fraction of the energy, and so less is lost in that release. So here we're going to remove another carbon dioxide. And just as before, whenever we remove the carbon dioxide, we can harvest some energy. And again, we're harvesting it in the form of a couple of electrons that are locked in NADH. We remove another carbon here and do the same thing. harvest a little more energy. All right, let me pause here to tell you what we've done. We started with 3 carbons in pyruvic acid, and we removed 1, 2, 3. We've removed them all, which means that the 4 carbons we have left are the 4 carbons with which we're going to restore this 4-carbon resident molecule. But this molecule here is not the same molecule as the 4-carbon resident because it's still holding some of the energy that we added to it when we added those 2 carbons. Remember, that energy was spread out through all the bonds of this molecule. So it still has some energy in it that we can harvest. And we're going to harvest it by rearranging those bonds. And when we do that harvest, we get an ATP, another electron carrier that's like NADH called FADH2, and one more NADH. Okay. When we've taken all that energy out, we have our 4-carbon resident restored, and the whole cycle can repeat. So with that 1 pyruvic acid, we now have 3 carbon dioxides left. We have 1 ATP, 1 FADH2, and 4 NADHs. Look over the drawing and make sure that I've done my math right. Okay, so from a single pyruvic acid molecule, we're able to harvest quite a lot of energy, but not a lot in ATP. In fact, if you recall, let me scroll back up to one slide here. In order to take care of our entire glucose, we have to do 2 pyruvic acids. So I'm going to multiply everything by 2 in terms of our products. When we have 2 pyruvic acids, we end up with 6 carbons, 2 ATPs, 2 FADH2s, and 8 NADHs. Now that is quite a harvest of energy, but very little of it is usable by the cells. The only usable part by the cells are the 2 ATPs. The rest needs to be cashed in to make more ATPs. And that cashing in happens in the 3rd and final step of the cellular respiration, which is called electron transport.